So the title of my talk is the U.S. military's role in biodiversity conservation. And although the military overall does a great job of managing threatened and endangered species, I'm going to be focusing mostly on the Department of Defense because that's who I work for. I'm a research biologist for the U.S. Army Corps of Engineers. So in general, the DoD administers about 25 million acres of land that's largely protected from development across more than 400 different military installations. So in general, military installations can actually be great places for wildlife because there's large, expansive, untapped areas that are free from fragmentation, habitat removal, etc. And not quite all the installations, but about 380 of them or so are considered to have significant cultural or natural resources. And that's because a lot of these areas are biodiversity hotspots. So places like Hawaii, California, the southeastern United States, Appalachia, and so on. One of the downsides, this is that there's a higher density of federally protected species than any other federal land management agency when we look at the DoD's properties as a whole. This figure is a little bit old now, but the general pattern still holds where the DoD there compared to, say, the Bureau of Land Management, Forest Service, Fish and Wildlife Service lands, National Park Service lands, whether we're talking about actually federally listed species under the Endangered Species Act, or just species that are of conservation concern, that number of species per 100,000 hectares, so the density there is higher on DoD lands than any of those other federal land management agencies. And since I work for the Army, this figure here also from this 2008 paper illustrates that the Army lands have a higher percentage of either listed or conservation concerned species than some of the other Department of Defense branches like the Air Force, Marine Corps, or the Navy. So where are all of these threatened and endangered species at? Well, a lot of them are in Hawaii. Again, that's a biodiversity hotspot. Guam has a couple with a high number of listed species. Again, the Southeast, like Cape Canaveral, down in Florida. California out west also has a high number of species. So again, the next few slides I'm going to show are some somewhat older data, but again, the general patterns I'm presenting here are still the same. So when you break it down by the different taxonomic groups of where a lot of money is spent on some of these conservation concern or listed species, it totals up pretty quickly when you get all this taxonomic diversity in there. And just over $1 billion alone was spent from the early 90s to the mid 2000s of the decade that we're currently in. So what is all this money spent on? Endangered species management is expensive. So what is it all spent on? Or land management in general is expensive. So what is it spent on? It should be familiar for the Floridians, things like prescribed fire, a common land management technique in those fire adapted ecosystems. So that's an expensive endeavor, but in most cases it's worth it for managing habitat and species. Surveys, monitoring, and inventory are a big expenditure, but again, almost an invaluable thing because monitoring these species over the long term is great for looking at population trends and so forth. But at a minimum, a lot of these installations need to know, do any listed or conservation concerned species occur on their lands? And if so, where are they? How many might there be, etc. Species reintroductions or augmenting imperiled populations through captive breeding and release of captive reared animals back onto the wild is a considerable expense, but is done on a lot of military installations. And I'll talk about some more of that later with a specific case study that I led. Invasive species management is another big one. So for example, feral hogs in the Southeast are a big, expensive problem. Bird airstrike hazard, or BASH, for the military loves acronym. So BASH is the acronym for bird airstrike hazard, causes a lot of damage and can even take human lives. And so there's been well over $1 billion in damages reported from airstrike from birds. So that's another huge expense. Ultimately, all of this money is spent to try to reduce conflict between species management and the military executing its mission, whether that be via land management, training, etc. Okay, so we talked about what all the money is spent on, but who is the money spent on? The red cockaded woodpecker is been pretty much the poster child for endangered species management for the Department of Defense and in the military in general. A lot of money has been spent on red cockaded woodpecker management. It's because a lot of installations in the Southeast occur on the longleaf pine habitat that these birds really like for nesting in particular. But the desert tortoise out west is not far behind. And you look at these lists here, mostly a lot of rare birds, whales, etc. Cost a lot of money. And there's the desert tortoise. So sometimes the species management can be viewed in a good light, where things, because of military installations, are probably one of the main reasons that the red cockaded woodpecker can persist in today's day and age. Sometimes it can be a little more contentious. The desert tortoise out on a few installations out west has been seen by many as sort of problematic and in the way of the military's mission. So there's definitely this trade-off between the military getting their way, what they need to do for training and so on, versus managing these species. So not that people don't care out of the goodness of their heart to want to conserve these species and manage them properly, but in many cases, they just have to. So there's a lot of regulatory drivers that necessitate all of these things that I've talked about, and that leads to these great expenses. The one that's predominantly relevant to me and my job is the Endangered Species Act, enacted in 1973. As already mentioned, there's about 400, if I say TESS throughout the rest of the talk, that's just referring to the acronym for threatened and endangered species. And so a lot of these species occur directly on the installations or places adjacent to them where they might transient onto an installation, or just general properties where military activities occur. And so simply by being present on an installation, in a training area, what have you, now the military might not be able to execute its mission. You have a nesting pair of red cockaded woodpeckers in a forest patch, and if military training might interfere with that, then you might have a problem. The Sykes Act of 1960 is another regulatory driver that's important for the Department of Defense, and it requires military installations to have what's called an INRAMP, or an Integrated Natural Resource Management Plan. So not every installation has an INRAMP, but many of them do. And it doesn't just apply to the actual listed species under the Endangered Species Act. It applies to other things, migratory birds that might not have full Endangered Species Act protection, fisheries, managing invasive species, and so on. A few more regulatory drivers, just mentioned migratory birds. So the Migratory Bird Treaty Act is another regulatory driver. About 1,000 species are listed under that. The Marine Mammal Protection Act of 1972 applies as well. And then NEPA, the National Environmental Policy Act of 1970, which requires federal agencies to assess potential impacts, environmental impacts of some of the things that they might be wanting to do on military lands. Okay, so enough of that legal mumbo jumbo. I'm going to spend the rest of the talk just talking about some of the particular projects that we've done in recent times. And when I say we, I might not have directly participated in some of the work that I'll be presenting, so I just want to give credit where credit's due. Another acronym for you here, though, ERDC. What is that? So within the DoD, I work for the United States Army, and within the Army is the Engineer Research and Development Center, or ERDC, which has 7 labs across mostly the eastern US. The headquarters of ERDC are down in Vicksburg, Mississippi, where 4 of those labs are. There's also a lab up in New Hampshire. There's a geospatial lab over in Virginia. But I work from the lab that's based in Champaign, Illinois, so East Central Illinois. And then there's some field offices scattered across the country as well. So CERL is the Construction Engineering Research Lab that I work for. So we'll start with some of these case studies pretty simply. Again, like I said, basic planning level baseline surveys are very important for these installations just to get an updated idea of what species occur on their lands and where do they occur. So on Fort Buchanan, Puerto Rico, one of my team members, Pat Wolf, he led a study that was published in 2018 where those red dots you can see on the map, there are point count stations. So they would go out and stand at one of these stations and listen and look for birds across the installation and record all the different birds that they saw. And they actually found that there was quite a diverse community, so about 60 species of native and some non-native birds as well. And one of their major findings was that these native birds were thriving pretty well in urban environments. If you look at the map here, this kind of grayish color is for urban areas on this map. So a lot of the installation is urbanized. But conserving these forest patches, so these green blotches here on the map, was also important for their persistence. But it's pretty easy to survey for these birds across Fort Buchanan because a lot of the installation is accessible. What about areas where you cannot survey for animals, in this case, birds? A lot of the times on military installations, huge training firing ranges, impact areas are just not accessible by people. But sometimes these threatened and endangered species do go into them. So this was a clever study done a while ago where they put an acoustic monitor on a balloon and could float it through some of these big inaccessible areas to remotely monitor these different sounds from the calling birds and then analyze the calls to see what species were occurring in those inaccessible areas. And it's not just birds that you can use these acoustic monitors for. You can do it with bats. You can get some of those ultrasonic high frequency calls, put some of these loggers out around a forest patch, see what kind of bats are occurring there. This acoustic device you can see here is actually used for frogs. So sometimes we'll refer to these things as frog loggers. And so Becky mentioned that we would go out to different stops around the Naples area and get out, listen for 5 minutes, see what frogs were calling, get back in Becky's van and go to the next spot. The nice thing is with these frog loggers, they're just out there recording data at whatever interval that you set. And then you can use the information to download all the different sound bites into some software, and you get what's called a spectrogram. So here, if you were just dying to know what a bullfrog spectrogram looks like, well, here's one. And these visually look different between the different species of calling frogs. And one of the great things about them is, so for example, this was from a published study in 2000, that these frog loggers are recording information at times when people typically wouldn't be out listening for them themselves. I would imagine most people are not out from midnight to about 6 in the morning listening to frogs. So the nice thing is the frog loggers are out there doing the listening for you. So we're getting good data that we maybe wouldn't get otherwise just by traditional methods of going out and listening with our ears. Okay, so SPAM. This is another remote, kind of newer acoustic monitoring method for frogs. And so I don't mean like the canned ham or your email getting cluttered up. What SPAM in this case stands for is Subsurface Passive Acoustic Monitoring. And what you can do is take this box there tied to a tree with a cable going down into the water, has a hydrophone on it. So it's a microphone that's actually underwater. And because many species of frogs, in addition to calling on land or being kind of half in, half out of the water, will call mainly underwater during breeding times. So if you were just out there, even with a frog logger on the tree, getting airborne sounds, going out listening yourself, you might miss some of these frogs that are calling underwater. And for comparative purposes, you can put a microphone on the surface as well to kind of see what's the level of intensity of calling at a given wetland for the frogs above and below the water. Another cool kind of vocal related project that was done in the past few years is this technique called conspecific attraction, or attracting members of the same species. And this works really well for vocalizing animals like frogs and birds. So this is very pertinent to the military because if you have, let's say, a group of frogs or birds that occur in some area, and then there's plan to maybe demolish that habitat to make a new training range or something like that. Well, you could just do it and accept that you might have some loss of those individuals. You could try to go out and capture them and translocate them and release them to a different area. Or you could be a little lazier about it and you could try to get them to move themselves. And this is where this conspecific attraction and the notion of that comes into play. So it's pretty simplistic in execution. You can just put a speaker connected to a battery so it'll last out in the field. And you can broadcast the call of your target organism in the area that you want them to come to. And the animals will in theory hear it and be attracted to it and settle in a new spot. And so this was tested out for bobwhite quail on Fort Polk, Louisiana, and gray tree frogs on Fort McCoy in Wisconsin, among some other species, but it seemed to work particularly well for these couple species. So in the case of the bobwhites, this project was done over 3 different years, and they did this work in 2 different ways. One scenario was in years 1 and 2, so we're looking at the left side of that 3D figure there. In years 1 and 2, there was no call broadcast at the sites. And in the 3rd year, they broadcast calls and had a much higher proportion of settlement at those sites. That's great, but to show it worked even better, because we don't just want the animals to come, we want them to stay. And so on the right side of the graph there, you can see that in the 1st year, no playback at those sites, broadcast the calls in the 2nd year, didn't do it in the 3rd year, and the settlement was still higher than it was in the 1st year, even in the absence of playback, which suggests that the birds actually, with 1 year's worth of broadcasting calls, that convinced them enough to stay on that site. It was pretty cool. Similar story for the gray tree frogs in Wisconsin. The researchers dug holes and put the big black plastic ponds that you might put in your garden at home. And some of them had calls broadcast near them and some of them did not. None of the ponds that did not have calls broadcast were settled, whereas about 40% of the ponds that did have calls broadcast from them were settled by the frogs. Not only did they come to them, but they actually used those fake ponds to breed in, which was awesome. Another technique we use quite a bit is game cameras. These are, again, you can go out and look for animals walking around the woods all day, and that can be useful. But sometimes it's nicer to just put something out there and let it do the surveying for you. And I see all the time when Becky makes her great posts on the preserve's Facebook page of all the different critters cruising around the preserve that are captured by the game camera images. And we do very similar work. Sometimes though, installations that we work on, some of them are huge and we have lots of cameras out there taking thousands and thousands and thousands of pictures across the course of a field season. And so what typically ends up happening is we hire some poor undergraduate student who goes through them by hand and sorts out some of the spurious ones that did not have an actual animal in it. Might just be plants waving around or something that flagged the camera to trigger to go off, and separate those from the images that actually have animals in them. But more recently, there was a machine learning algorithm that was developed where you can actually essentially automate the process of processing these trail camera images. And so you can use a program and train the algorithm. And the more you train it, the better it gets. And not only can this algorithm actually identify species, which is cool enough on its own, it gets way more detailed than that in some instances. You can, with enough training data, the program can actually detect, is the animal doing something like walking? Is it lying down? Is it foraging? Maybe sometimes age classes. And so you have to go through by hand and tell it first, but over enough time, you can get some very, very valuable information just beyond presence absence of your target species. It is somewhat in its infancy still, and it is imperfect and it doesn't work for everything. So here's an example of that. At Fort Belvoir, Virginia, there was this huge retention wall here that kind of grate across it and all those rocks. And we were doing some reptile and amphibian surveys on this base. And in the spring, we noticed rat snakes were emerging from this rock wall. So they were probably overwintering inside of the rocks. So we put up a camera on the rock wall to try to get pictures of them emerging in the springtime. And then I ran about 8,000 or so images through one of those automated classification software programs, which it works basically on pixel changes from one image to the next. So the red boxes on the screen there is where the program thought based on pixel changes an animal was. What it really was just as the light was changing throughout the day, that's what the software is picking up on. So there is a snake in this picture, and I'll give you a few seconds if you haven't already spotted it to find it, just to illustrate how sometimes all the fancy technology and software and such out there, that's great and it's very useful and it can save a lot of time and effort, but sometimes just your own eyes are more trustworthy. So hopefully you've had enough time maybe to look for it, but if you haven't found it, there it is up in the top right corner there. There's a rat snake kind of weaving in and out of the grate, getting some sun before he decides to crawl out and go start his year up in the woods. Environmental DNA, or eDNA for short, is another huge boom in this kind of work for species detection. So there I'm holding a Louisiana pine snake, which is thought to be one of the rarest snakes in North America. And they occur in this longleaf pine habitat, similar to the red cockaded woodpecker, which historically there was a lot of loss of that kind of habitat, which is one of the reasons that is believed for the snake's decline. And the snakes spend a lot of their time down in these pocket gopher burrows. Not only do the pocket gophers provide food for the snakes, but they also serve as a nice refuge to escape the elements. So they spend more than not of their time actually underground inside of these pocket gopher complexes. So detecting the snake historically is very hard. Oftentimes you can go out and drive roads and just hope you encounter a snake crossing a road. You can put out traps, which doesn't really, it works great for some species, not so much for pine snakes. You can go out and visually survey for them, but I've been down at these sites in the middle of August, in the middle of the day. It's not very pleasant. So if we want to get a better idea of the snake's distribution across, in this case, Fort Polk, Louisiana, what if we just took some dirt from these pocket gopher mounds and test them for the pine snake's DNA? And it turns out it works, and it works really well. So this is more of a pilot study here that was done in areas where we knew that the snakes were hanging out, but now this year we're going to expand this project across the whole base and try to get a really good understanding of the snake's distribution using this eDNA technique. And it's not just for terrestrial animals like pine snakes. We can actually do this for aquatic animals as well. So also on Fort Polk, there's a lot of river and creek systems that go throughout the base. And we have tested water samples from a lot of these sites for endangered crayfish or the pretty rare and elusive alligator snapping turtle. And in the case of the snapping turtle, it works really well. We've actually found some new sites where the turtles were not known to occur previously based on using eDNA. So the installation biologists love this because in their eyes, you can just go out and collect some dirt or dunk a bottle in the water, and then magically we will let them know, yep, we found the target organism's DNA or we didn't. But there's a lot of work on the back end, a lot of laboratory work to extract the DNA from these samples, to run the analysis, to understand, are we actually detecting the species DNA or not? Are we getting false positives, false negatives, et cetera? So there's a lot of work that goes into it, but it's a pretty cool developing science that holds a lot of promise for understanding species presence absence in the future. Okay, so this was part of my master's thesis research that was done up on Camp Grayling in northern Michigan. And up there, there seems to be a pretty robust population of massasauga rattlesnakes, which were listed as threatened under the Endangered Species Act in 2016. So Camp Grayling has had a long interest in understanding what habitats are the snakes using, where do they go on the installation, how might we reduce conflict between soldiers encountering the snakes and letting the rattlesnakes persist. So we use radio telemetry to do this, where we implant that bright oblong kind of Tootsie Roll shaped object there with this little wavy whip-like structure. That's a radio transmitter and an antenna that we surgically implant inside the snake's body, sew them up, let them heal for a while, and then release them back out into the wild. And then we start tracking them with a receiver unit and an antenna. So the receiver, you punch in the unique code or the unique frequency for each rattlesnake's transmitter. So instead of tuning in your favorite radio station in your car, you're tuning in your favorite rattlesnake to go find out where they've gone. And actually move a surprising distance for a relatively small animal. And then you use the antenna, which my, at the time, girlfriend and now wife and mother of my children was helping me in the field track the rattlesnakes. It's kind of like this big game of hide and seek where all summer long, the snakes go hide and we find them, and then they go hide again, and then we go find them again 2 to 3 times a week. So hopefully you can hear that there. That's what the beeping sounds like. So a few things about that. Why do we have the receiver off of the antenna? Well, the snake was still underground at this time for the winter. This was early spring when they're starting to emerge from their overwintering sites. And you may have noticed, if you could hear that the sound was getting louder and fainter. That's because the closer you are to the transmitter and the animal, then the louder the beeping gets. And in this case, our transmitters were actually temperature sensitive. So the warmer the animal was inside its body holding that temperature sensitive transmitter, the faster and louder it beeps. In this case, it was pretty slow because the snake's underground and temperatures weren't that much above freezing at that point. You do have to be careful when tracking rattlesnakes for some obvious reasons, one of which, here's another probably easier find the snake picture. When we'd be out in the field tracking the rattlesnakes, we would take environmental data such as the air temperature, the habitat the snake was in, maybe some measurements of the vegetation around the snake. But then we would also go out and take data at a random point to kind of see, just compared to any old place we went to and recorded the same information, how did that randomly differ from the habitat that the snakes were selecting? So I would set my antenna down, go off to take my random data and come back and go to pick up my antenna, and there's a coil of rattlesnake that was not there when I set the antenna down to go take the random data. So we do a lot of tracking with the snakes by hand, but some more recent technology is where we can actually use automated receiving telemetry units to get information about the snakes on the landscape at any given time. So we construct these big towers that have 6 antennas instead of just the one that we were holding by hand. You install those antennas on top of the tower. And then at the bottom of the tower is a receiving unit where you program in each snake's frequency and set it to cycle however often you want to search for the snake's frequency on the air. So in this case, we did about every 5 minutes. We had it cycle through, did a tower, we had a few of these across the landscape, did a tower detect a snake or not? And that's great because that gives us information such as things like movement frequency. So across any given day, often are the snakes moving on average throughout the course of the day. So this shows a nice kind of pattern that these snakes are predominantly diurnal. So they're active mostly in the peak of the day, and then their activity kind of slows down again towards nighttime because even in the peak of summer up in northern Michigan, it still gets pretty cool. And then their activity picks up again the next morning. So we can look at their activity patterns with these automated receiving units across the day. We can also look at it across the course of the season when they're active. So the snakes typically emerge from overwintering around April, and then their activity picks up big time going into June, July, and August. They're going out foraging, mating, doing things that snakes need to do. And then they slowly return to their overwintering sites as fall sets in September. And then come October and November, they park it in their overwintering site and spend the rest of the winter there and do it all again next spring. The overwintering phenology of the snakes is pretty fascinating. So you'd think that it'd be kind of boring because the snakes just go underground and then sit there, and then we just wait until they come out to study the interesting parts when they're active in the springtime, right? Well, it's actually really cool with their wintering behavior. We can use these little borescopes here to put down inside their overwintering site, which in some places is something like a crayfish burrow. Up here in northern Michigan, any kind of root or stump systems, mammal burrows, anywhere where they can really get down and get into the groundwater is important for them. And usually they are not alone. So in this case alone, these snakes were coming up towards the surface, getting ready to emerge from their overwintering site for the season. And there's at least 1, 2, 3, even the bottom underneath the snake with looks like at least 2, might be another nose and eye there. So there's maybe at least 4 rattlesnakes, maybe more. We've seen up to 6 or more inside of a single burrow together. So they kind of huddle up together for the winter time and then disperse in the spring. We've done projects in the past where we monitor the temperature and humidity of the air in the soil. I think the massasaugas here are a great example of how to balance species management with land management practices. So I talked earlier about prescribed fire. A lot of the habitat massasaugas occurs in is dependent historically on fire. And so land managers, whether it's for actual species management or for land management to, for example, keep a firing range open, they need to burn them quite often. The problem is if you burn at the wrong time, you might have snakes that are sluggish coming out from their overwintering sites that are very susceptible to getting burned up by fire. So could we use temperature to predict when the snakes are going to emerge? And if so, maybe you should burn before that happens as opposed to after. So Chad Smith, during his master's thesis with the same population of snakes up in northern Michigan, had temperature loggers at varying depths down into the soil. So from about 10 centimeters, almost half a meter down, and track the temperatures throughout the course of the time when the snakes are typically emerging. So that's signified by these 2 red bars here. This is the emergence period of when they're coming out. And what happens is this blue line, the temperature at closer to the surface is the coldest, and the deeper temperature is the warmest in early spring, late winter. Then there starts to be this flip. There's this thermal inversion where now the temperature is the warmest at the surface. And this is thought to be the kind of trigger that promotes the snakes to come out for the ground. So if you're going to burn, do it before you get to this first red line here, because this is when the snakes are still underground. And it seemed to match up pretty well with the thermal properties of the soil. Okay, that was snakes. If you don't like snakes, sorry, I'm shifting gears now to something more cute and cuddly, not mammals, but turtles. This was part of my PhD research that I did at Fort Custer, which is down by Battle Creek in southern Michigan. And I talked earlier about reintroducing species and captive rearing species for release onto military lands as an expensive endeavor. And it's true, and not just with military lands, but in general, this is a common practice called head starting or translocation. And it's intuitive. You raise up baby animals past some point where they're very susceptible to predation. Baby turtles, most of them don't make it out of the egg. And so if you can raise them safely in captivity to a size where they will be larger and less susceptible to predation, then hopefully that will help the population out in the wild. And so that's what Fort Custer wanted to figure out. So we collected some eggs from naturally laid nests at Fort Custer, and then we hatched them in captivity, and then we split them into 2 different rearing treatments. The treatment on the left there, we called our control or our unenriched group, where they were raised very simply. And this is oftentimes how you will see animals head started for release into the wild, how they're raised. It's cheap, it's easy to feed and clean them. You can go through and you can service a bunch of cages at one time. But does that come at a cost to their development as they're being reared in captivity? Does it come at cost to growth or their behavior? Because what we want them to do is be well prepared for release into the wild, and we want them to survive. That's the ultimate goal. So on the right side is what we called our enriched group. Probably didn't have to differentiate the 2 groups there, but it's pretty obvious that they have a better setup. Multiple places to hide, soak in the water, bury in the substrate, plants to hide under. There were 4 turtles in each of those tubs, so they had some social interaction. And so overall, we thought this would be the better way to raise the turtles that would prepare them for the wild. So we had them for up to 2 years in captivity. Some of them in each group were released after about a year of being head started. Then the rest of them we held on to until they were almost 2 years old and released them. So during that time, we took good care of them, tracked their growth rates, measured their behavior through a few different experiments, and so on. And we didn't just say, hey, have a good life. You're not going to get cared by us anymore. We didn't just dump them out into the wild. We actually did what's called a soft release. So we built these enclosures out in the forest paths where the turtles were released, and we let them spend about a month inside of these enclosures so they could get used to the elements outside, hopefully acclimate them, soften the transition between captivity and being released into nature. And then we just cut the pen open, let them exit at will. We had radio transmitters on the turtles that we epoxied to their shells, similar to the rattlesnakes, but those ones were internally planted. These ones were just glued on their shell. And same thing, we followed them around using radio telemetry to see where they were going, what they were doing, and how they were surviving. We looked at various things when we were tracking the turtles, but ultimately we want to know, out of these 4 groups, you could call them, whether you were raised in an enriched or unenriched environment, and if you were released after about 1 year in captivity or 2 years in captivity, which group did best overall with survival. So our first year on the left side of the left panel there in our 2017 release cohort, at first it looked like our enriched turtles were doing well. We had an immediate drop off of our unenriched turtles, the ones raised in that simplistic environment. And it looked like our turtles that were raised in the more naturalistic environment were doing well for several weeks after they were released. But then we had some die off of them as well. And so we actually had equal survival in the first year at the end of the season when they start to go underground to overwinter as well. In our second year cohort, so now these turtles that are older and larger that were raised in captivity longer, our enriched turtles, contrary to what we thought was going to happen, actually has a lower survival, whereas our unenriched turtles had the highest survival of any of the 3 groups, about 60% survival overall. Well, that's not what we thought was going to happen. We thought that raising the turtles in a naturalistic environment would better prepare them for the wild. So why did these turtles that were raised in a simplistic condition where they didn't really have much stimulation, why did they do better? Well, we think that's related to their body size. The turtles that were raised in those naturalistic tubs maybe had to compete for food because there was multiple turtles per tub. Maybe they sacrificed foraging a lot because we would scatter their food around their tubs. So we made it more challenging for them to forage. Maybe they just couldn't forage as well. Maybe they wanted to spend more time hiding versus eating. Whereas our unenriched turtles in that simplistic condition, all they really had to do was just sit there and eat and grow. And so ultimately that seemed to be what prompted the higher survival. Keep them in captivity longer so they could grow bigger, and the biggest turtles seemed to do the best, as shown here. So at a certain point, once our turtles reached about 60 grams or so, they had almost 100% survival. So that might be a kind of cutoff threshold for, if you're going to release these baby box turtles into the wild, you want to get them to this minimum size before releasing them because their chances for survival are increased. We wanted to know, we knew we were going to lose some turtles. It's just the reality of releasing baby animals into the wild. We knew that some of them were going to get eaten. And so every time we tracked the turtle, we had a camera, one of those game cameras I was talking about earlier, on a tripod. And we would point it right a few meters back from where the turtle was situated and focus the camera on the area where the turtle was. And it sounds kind of morbid, but in hopes of getting on film what was eating them, because we don't really know. We have ideas about what eats baby turtles in the wild, but we don't have a great idea. Unfortunately, we didn't get a single predation event on film, despite losing lots of turtles to predators. We would just find this. We would find a mangled, chewed up shell, and if we were lucky, the transmitter was there with it. But this is as close as we ever got to identifying what maybe predators were eating these baby turtles. So I thought, okay, I've got to sort of figure out a way to get to the bottom of this. We need to know if we're going to keep releasing turtles into the wild, and if their main cause of death is being eaten, we should get a better idea of what's eating them. So that's where I had the idea to put models out in the wild. You can probably guess which one is the real turtle there and which one isn't. And I was looking around at different toy distributor websites, so on. I couldn't find a good model that I really liked. Sometimes people, for these model predation studies, make things out of clay or plasticine, like silicone, but I didn't feel like doing that. So we actually, at the University of Illinois, in our business school, had a 3D printing lab. And I found this website called Thingiverse, where there are pre-existing designs for 3D printed objects. And I found a turtle that to me looked good enough to be a box turtle. And so I went to the lab there and said, could you print these up? And they said, yeah, it'd be no problem. So for about $5 a piece, they printed up these turtles for us. And we started putting them around the landscape at a few different sites. One was up at Fort Custer where the head starting turtle work took place. And then we did it at a couple different sites around the Champaign area in Illinois. And so similar to how we were tracking the turtles by putting the cameras on them, this time we just put the turtles out there, the plastic turtles, to see maybe what was going after them. Not to our surprise, raccoons were by far and away the most important predator of the model. So here's a raccoon going after one of our 3D printed turtles there, and gives it an honest effort, and then eventually decides, okay, this is probably not real, and gives up. But it wasn't just raccoons. We actually had other animals that were seemingly attacking these models. So maybe to your surprise, things like chipmunks and squirrels were going after these models, which aren't they herbivores? Well, no, actually some of these rodents can be rather omnivorous. And for example, squirrels are really well known for going up into trees and eating entire clutches of bird eggs or eating nestlings out of bird nests. So they can be pretty voracious at times. But in addition to the raccoons, other mesopredators like possums were actually going after the models as well. Since we had cameras on them, we could actually see what time of day these different predator types, whether it was a raccoon or a possum, those mesopredators, or those chipmunks and squirrels are rodents, what time of day they were going after the models and how often. And so this actually reflected the natural behavior of those predator groups rather well. The rodents didn't attack them as much and were pretty diurnal. So during the daytime, we're going after the models. And then in the evening and through the nighttime is when the mesopredators were going after them the most, particularly around 8 o'clock. So if you were a baby turtle and you were out after Jeopardy was on, you were pretty much screwed, it seemed like. So what I want to mainly point out in this figure is this dashed blue line here that shows that the likelihood of a turtle being attacked by a raccoon decreased as the height of the vegetation around them it was increasing. And so basically the more dense the vegetation was around the model, the less likely that the raccoons were to find it. So that kind of comports well with the natural history of the baby turtles. Hide from predators, you might not be as likely to be detected and thus eaten. For this figure here, let's focus on this red line, which was the probability of a rodent detecting a model based on how far they were from some sort of woody structure, like a log or a slash pile or a bramble, something like that. So even though the probability was never very high to begin with, it dropped off to nothing once the models were farther from things like logs. And that's probably reflected also in the behavior of most rodents who use things like fallen logs and such as little runways when they're out and about in the woods. And so it's not just animals that we study as part of our jobs. We also occasionally do some plant work. So this is an example of a biological assessment that we worked on for Oahu. We're also doing one right now for the island of Hawaii, where we are investigating and doing consulting with these local Army bases to try to figure out what is the risk from the different activities that they do on the installation, how would that affect some of these threatened and endangered species. And so things like weedy encroachment, invasive plants, fire, etc. What's the risk from those activities to these listed plant species? Okay, I'll talk for just a brief minute about some of the different organizations that are involved in the Department of Defense and military when it comes to species surveying, inventory, management, so on. One of them is the DoD PARC, the Partners in Amphibian and Reptile Conservation. They are big proponents for these reptile species and do a great job with things like public outreach, getting people exposed to some of these less loved critters. They have a great series of fact sheets based on the different, in this case, snakes that occur on military installations, where they occur, what kind of habitat, so on and so forth. So great series of those fact sheets. They do things like safety training, so soldiers can learn how to safely remove venomous snakes from something like a training area. The DoD Partners in Flight is essentially the bird version of the DoD PARC. And so again, they do a lot of great work with conserving some of the avian species that occur on DoD lands. One of their main focuses is to focus on reducing BASH, that bird airstrike hazard, and do a great job of that. Bats are another one. There's Bat Conservation International that does a lot of work with the military to help manage and conserve bats. One of the big things right now, the right picture here is showing what looks to be a bat infected with what's been known as white nose syndrome. It's a fungal pathogen that can spread through entire colonies of bats and wipe them out. And so organizations like BCI here are doing a great job to try to unravel some of those mysteries around that to conserve bats. There's also, just for another example here, an Armed Forces Pest Management Board. Things like ticks spreading across some of their historically not native ranges and so on. It happens when you have people moving around from base to base. So there's just another example of some organizations that are doing these sort of things in the armed forces. So sort of wrap up here, this is just an example of everything I've talked about. This is ultimately what it's all about. All the work that we put in is to try to get these species, if they are listed under the Endangered Species Act, to get them off of the list. When a species is listed, the goal is not to keep them on there forever. It's to get them off of the list as soon as realistically possible. And so in this case, a few years ago, the black cat vireo that was, or is, an inhabitant of a large base in Texas, Fort Hood, where we do a lot of work, the bird was actually delisted. So just an example of a conservation success story. So to wrap up here, the Department of Defense has diverse challenges for solving environmental problems while simultaneously making sure that they achieve their mission. There's an ever-evolving need for effective methods to monitor, inventory, detect species, and make sure that they persist on these lands, which requires collaboration from a lot of different people. From active duty personnel play a large role in it. People who are Army civilians, that's our job, we're Army civilians, and numerous stakeholders as well. So with that, I'll take some questions if you have any, and thank you for your time listening, and thanks again to Becky for inviting me to give the talk to you, and I hope you enjoyed, and I will open it up now for questions. Now I'm just looking at the chat here. The battery life of an implanted transmitter. So I'll go to that question first. It depends. And so really it depends on the size of the transmitter, and that depends on the size of your organism that you're studying. So a lot of the times, like for the massasaugas, we could put a transmitter in them that was only about 5 grams in weight. And those tended to last between a year or two. Some bigger snakes, you can put a much heavier transmitter that can last 2 to 3 years. In the case of our little turtles, that the transmitter weighed half a gram or so, some of them a gram. They had to considerably slow the pulse rate of the transmitter down to actually get them to last more than just a few weeks. But ordinarily, those sometimes might only last weeks, so you have to replace them pretty often. Any thoughts on how broadly applicable is the eDNA approach? That's a really good question, because like I said, the installation managers love it. They love the idea that you can just go out and collect an environmental sample and then magically get DNA out of it. But in reality, it's not that simple. And so some organisms, it seems to just work better for than others. Why we think it works pretty well for the pine snakes is because they spend a lot of time underground just sitting there shedding DNA. And inside those pocket gopher burrows, The DNA is protected from heat, from sunlight, like UV radiation will break down the DNA. And so it can persist for quite a long time, we think. We're actually doing some field experiments this summer where we're putting synthetic DNA of the pine snakes into those burrows, and then we'll continually sample those to see how repeatedly and how long we can actually get eDNA detections from those. Other species, it doesn't seem to work that well for. So for some turtle species, it's been found that you have to take a sample almost right from where the turtle was occupied recently to be able to get a detection of it, just through some field experiments to figure that out. So for some reptiles, it might not work too well. Things that shed DNA a lot, like amphibians that don't have quite that hard integument like scales or a shell like a reptile, seem to be pretty good candidates for it. Insects seem to be another pretty good one. A collaborator of mine's actually taking flowers and grinding those up and testing those for different pollinator eDNA. So it's a pretty powerful tool when used in the right context.
Sent the plastic decoys out in the field?
Yeah, we did. That's a great question. So we actually just took water that our captive turtles had been soaking in, and we stored them in these vials that did not allow air to escape from them, and just drew it out with a syringe right at the moment we were putting the model out in the field. And then so we used that as like our scented treatment. And then the control for that was just putting tap water on a model. And so we actually did it sort of a split experimental design where we had the scent treatment, but we also had an exposure treatment. So some of our turtles, we buried them quite a bit. So just some of them were like the back of the turtle was exposed, and other ones were just sitting on the surface. And so we were just trying to see, does the scent and the exposure, does that matter? And for things like raccoons, it didn't really seem to matter. And that's probably reflected in that they use a lot of different senses to find food. But actually for the rodents, the exposed models were attacked more often than there are models that were buried a little bit, which probably just reflects them being a predominantly visual forage.
My eyes about a lot of things. I just wish we could do a lot of that here, especially the predation of the hatchlings. It would be great if I could do something like that here to just see what is getting. I know when the raccoons get them, because so your box turtles, did the raccoon eat the shell and everything, or what was left is what I'm asking.
Yeah, it's kind of hard to tell because we don't really know, because we track a turtle one day and then the next we come back and just find the transmitter. Sometimes, like the one picture I showed was a pretty intact shell. Other times we just find little fragments or scales from the carapace on the ground. Rarely will we find nothing at all. Some of them you could see little bite marks in the epoxy from the transmitter. So that makes me think some mesocarnivore, like a fox or raccoon or possum, whatever. Other times, based on the way the shell was chewed up and the fragments, makes me believe more so things like chipmunks. We're going after them a lot. And especially sometimes we find shell fragments and then little holes were kind of around the area, some fallen logs, and we see chipmunks scurrying back and forth. So we don't really know. Part of the problem is we could even take like the shells and test them for eDNA to see what's on there if we're getting saliva or something from a predator. Then the tricky thing becomes disentangling, well, is whatever we detect DNA-wise on there from the actual animal that ate the turtle, or is it from a scavenger? So, as far as I'm aware, that the 3D printed way is the closest we've gotten to getting any real robust idea of what some of these predators might be.
Yeah, see, yeah, I think you're showing people too, you really have to think outside the box. Things aren't as simple. You have to think all areas. And here what I found with the gopher tortoise hat, well, it goes up to 3-year-olds. When the raccoons get them, even the ones that are really little, they actually reach in and rip them out of the shell and don't damage the shell at all. And you know, on hatchlings, you know, ones that are under a year old especially, the shell's fairly soft. You would think they would just chew them, bite on them, but they rip them out of the shell.
Yeah, that's crazy. Because I've seen videos of raccoons going after adult box turtles, which can, our hatchlings couldn't close up their shell all the way. Some of them are pretty close, but that isn't really quite developed all the way yet by the time we were releasing them. But I've seen videos of nimble raccoons just trying to pry open a box turtle shell and have a hard time doing it. But they must get chewed on at some point, because I'd say maybe 1 out of every 3 or 4 females we find during the nesting season is missing a foot or something like that, which leads me to believe raccoons are chewing on them when they're nesting and pretty vulnerable. But yeah, that's crazy that they just rip them out all the way.
Yeah, a lot of our Florida box turtles that we have on the property, which are in a study, which we have a talk on at the end of March. So that'll be interesting to compare what you were finding and what Jordan's found. But a lot of them are chewed up. The adults are chewed up. Some have like canine holes in them.
Rough life for a turtle.
Yeah.